Yes, very good morning, everyone.
Morning, sir.
Morning, sir.
Welcome
morning,
to
sir.
today's session. Today's, with respect to previous yesterday's continuation by Mr. Viresh Kumar. We just wait few more minutes for Seth to join, okay? Once he joins, we can start the class. Thank you.
Let me start.
So good morning, sir. Now we can continue, I guess.
Yeah. Okay. So good morning, everybody. We started the session on the autoclave, so we are going to continue a part of that today, covering the types of autoclaves and then also looking at the EN 285 specifications or guidelines for autoclaves. So we did discuss yesterday in brief about the geometrical configuration of autoclave and how they are classified as horizontal, vertical, double door autoclaves, single door autoclaves, etc. But let us get into the technical classification that is used first to define the autoclaves based on the type of the autoclave itself, which is primarily a dependency on the function. So there are different type of autoclaves which are present in the market, and there are four major criteria based on which these are classified. The first one is a laboratory bench autoclave, or what is called as an N-type autoclave. These are nothing but domestic systems or pressure cooker types systems which are still used in many parts of the world. As I said, in smaller clinics and smaller cities, these are still in use. The most modern type has a metal chamber with a secure metal lead that can be fastened and sealed with a rubber gasket. It has an air and steam discharge tap, pressure gauge, safety valve. There is an electrical immersion heater at the bottom of the chamber. So it is something which looks like this, and that's the type of an autoclave which is N-type autoclave. The second one is a gravity displacement type autoclave. This is the common type of autoclave which is used in most of the modern laboratories, and is available now as a standard equipment from the various manufacturers. In this type of autoclave, the steam is created inside the chamber via a heating unit, which then moves around the chamber for sterilization. And this type of autoclaves are comparatively cheaper than the others. So again, when we go to this, most of these autoclaves are either of this kind of configuration, or they could be even extended up to this kind of configuration, which is in vertical autoclave. The modern version of it with the full automation and compliance is something that you see here. So these are the normal laboratory autoclaves which are present there, and this is called as a gravity displacement. Why is it called as a gravity displacement? Because you will inject or create steam and put it on the chamber from the top, and it will displace the air by gravity from the bottom. There'll be a drain connection to this chamber, and therefore you achieve the air removal followed by the sterilization. Positive pressure displacement type or a B type autoclave. In this type, the steam is generated in a separate steam generator. So you have a separate boiler in which the steam gets generated, which is then passed into the autoclave. The autoclave is faster as the steam can be generated within seconds. And this type of an autoclave is an improvement over the gravity displacement autoclave. So some of the autoclaves which are used even in the hospitals, or in, like for example, in a horizontal autoclave which we show here as a picture, the steam is generated outside. There is no vacuum system which is attached to this. It's just based on a positive displacement, but the steam there is generated externally. In all these systems, what happens is, like if you look at the pressure cooker type or the common laboratory autoclave, there will be a uh, 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 water bath or a water trough in which there will be water poured, and that is heated, which generates the steam in situ inside the chamber. In cases like the horizontal autoclave or the positive displacement autoclave that we are talking about, which is B type autoclave, we connect the steam source from outside the chamber, and therefore you are able to send the steam inside to the chamber. You don't have to worry about the water levels inside the chamber or anything. You just have to make sure that the steam at the right pressure and the right quality is injected into the chamber. These are very well, of course, systems which are used in the industry because of the fact that you have steam generators, the boilers are there, and therefore it is easier for you to just send that steam across, not to worry about water and level of the water. Because if you have to worry about the level of the water, the effective space within the chamber again would go down if you had to have a large chamber like this, and if you had to generate a water system. Now, it is not an uncommon practice that sometimes you also have a steam generator provided by the autoclave supplier as an accessory to this autoclave, but it would be a separate equipment where you maintain the water level, and then you heat up, you generate the steam and inject it into the system. In sterile manufacturing operations, however, we are only going to use what is called as a pure steam. So we did study about pure steam or the different type of waters that we use in the pharmaceutical industry. So a steam which is generated using purified water, which is on condensation going to be free of endotoxin, free of any gases, and is meeting the quality of water as specified by US Pharmacopoeia section 41, is what you will use in the modern in sterile manufacturing operations. So the pure steam generators are a separate equipment, independent equipment, which are available, which are kept in a central location, from where there is a steam line which is run to all the equipments which use steam. Connection is given to the autoclave, and generally at the autoclave junction you will have a pressure regulation valve. You will also have a point where you can collect the steam and check the quality from the perspective of moisture content and all that, and then you allow the steam inside the chamber from the top. So the chamber is generally injected from the top nozzles here, which allows the air to be displaced from a drain point at the bottom. And of course, you will also be doing a vacuum displacement. If you don't do a vacuum, then it would still, a chamber of this size can still be a positive pressure displacement type of a autoclave. So the key factor is that the steam is generated from outside. Now, That is a B type one. It is expensive as compared to gravity displacement type autoclave, because you have to now consider the cost of two equipments, and then you have certain construction features inside the equipment to make sure that there is a proper air displacement, etc., etc. And as a result of which, the positive pressure displacement type or the B type autoclaves are expensive as compared to gravity displacement type autoclaves. The negative pressure displacement type, or a S type, this is the most commonly used and recommended type of autoclave which you will use in the sterile manufacturing processes, or actually in most of the industrial applications. Here, the only addition to the positive pressure is you also have a vacuum generator attached to the system. You use the vacuum evacuation phase for removal of the air initially from the chamber, and then you add the steam, you do the pulsation of the vacuum to make sure that you have evacuated the air completely out of the system, and then you add in the steam and then carry out your sterilization program. This, of course, is got the best results in terms of efficiency of sterility assurance and sorry, and ensuring that it is accurate and consistent and repeatable. Undoubtedly, all the types of pressure cook, the autoclaves that we are talking about, whether it is a laboratory bench, gravity displacement, positive displacement, negative pressure displacement, are today available in modern industry with a lot of automation, with enough controls. So when you look at a laboratory autoclave like this, which is a vertical autoclave, has got a built-in automation system to measure the temperature, the water levels in this case, generate the steam accurately, make sure that the air gets removed. So the automation today has helped us in terms of making sure that even the autoclaves, whichever type you use, will give you a good control on your sterilization process. So you would ask me, why would somebody choose a most expensive type? So it is again dependent on what application you are going with. If you're going with a laboratory application, obviously you're not going to use a large autoclave of negative pressure. Yeah, you could use a vertical autoclave in its place. You could use a horizontal autoclave and combine it with vacuum if you wanted it even in the laboratories. But it is generally not required, because the cycles or the load is very, very small, and you are trying to sterilize it. It is easy to displace the air because the amount of air is not very high, and therefore it is easy for us to remove the air from the system on a very small equipment of this size. Now, when we talk about, say, microbiology lab, or when we talk about smaller laboratories where you're doing certain amount of experiments, or you're preparing, say, the inoculum, etc., again, a positive pressure could suffice your application, because again, the sizes of those autoclaves are not going to be very large. They're going to be in the region of 600 liters or 300 liters, and they sometimes are sufficient to meet your requirement. There are microbiology labs where you need larger autoclaves, and obviously there you would use a vacuum displacement. So it is dependent on the quantum of load, the quantum of the size of the autoclave, the type of the autoclave, etc., where you define which suits your purpose the best, and therefore you decide which autoclave you will buy. Now, this is one classification which is based on the type of autoclave. Now, there is another classification of the autoclave also, which is based on the application for which you use the autoclave. So what are the applications that you generally use this? The first one is for component preparation. So whether it is a sterile manufacturing process or microbiology lab and inoculum preparation lab, you need to, or in even in hospitals, for example, you need to sterilize the components which are used. The components could be media, the components could be garments, the components could be Tools and tackles components could be even things like rubber stoppers and could be certain steel containers, certain plastic containers which can withstand high temperatures and or moist temperatures, can be wrapped in a permeable bag like a Tyvek bag, and you use them. So when you have to use prepare the components which you use in the sterile manufacturing process or in any of your activity which where asepticity is important, then you would do what is called as a component preparation, and this is one application. So you are using steam sterilizer for component preparation. The type of autoclave you use will again depend, as we said, on the load and on the true on the need of the autoclave, and therefore you would either use a bench scale autoclave, or you would use a double door autoclave, or you could use a positive pressure, just a positive pressure. Now, this is of course used for all the components where it is compatible with moist heat based sterilization and is the most preferred method of sterilization. So the component preparation, this is one of the biggest applications of the autoclaves, and that is one classification. The second one is terminal sterilization. So when we do terminal sterilization of the products, and we've had a full session on terminal sterilization, and on that sterilization, when we look at it, the final container allows you to do a moist heat sterilization. Generally, the parenteral products which come in infusion bags are a big example of this. You use a steam sterilizer for terminal sterilization, and there is the air water mixture and the steam based system which we have studied in the past. So in that situation, this is the other application, that you're using a steam sterilizer for terminal sterilization of the product. The last application is decontamination. So in the laboratories, in the manufacturing plants, and especially into the biopharmaceutical industry, where you have a biological process step or a bioprocess step before you get into the purification processes, which could be physicochemical separations, or certain purifications involving chromatography, filtration, etc., or even in solvent-based process, you have the biological process where you have a lot of cultures that you're handling with, or a lot of components that you're handling it, and you need to decontaminate these because they have got a contamination, or they are already touching the biological or the microbial loads, including the culture actually with which you are working. They could be also, as we looked at yesterday, filtration of viruses using filters, right? Or microbes are also kind of filtered using filters, capsule filters or cartridge filters. For all these applications, after you've finished the activity, you need to destroy that microorganism, because you don't just want to leave it out, because that will spread into the atmosphere. Because if you leave it for long enough, there is the right condition, it will start to grow, and the spores will start to spread. So to prevent any of those contaminants to spread into the environment and contaminate other services nearby or the area in which it is being used, you have to decontaminate some of these using heat. And that's where you use a decontamination autoclave, which is also based on moist heat sterilization, and is effectively used to destroy all kinds of vegetative bacteria, bacterial spores, and viruses, apart from your media plates, etc. So in principle, these autoclaves are no different from your component preparation autoclaves and your horizontal autoclaves or vertical autoclaves that you saw. The difference there is that when we come across to the sterilization cycles or the controls that are required there, you are not looking at a sterility assurance there. You're just ensuring that there is sufficient kill, so that your microorganism is not in a phase of growth, and it is either in a death phase or it is in a lag phase, so that you can dispose it off, you can discard it out safely into a biomedical waste stream, or if it was a liquid, sometimes you even decontaminate liquid by heat, then you can send it to an effluent treatment plant where it gets broken down further. So steam sterilization is a process which is used for decontamination. Also, this is a very, very important process that for scientists who are working in the research and development area, for example, they are doing a lot of discoveries, and during those discoveries, they have a lot of these cultures which are being used, and they have done some discoveries. But of course, after they finish their experiment, they have to throw it out. But because they have done work, it's of a great importance in terms of the IP that it has with it, because you have done some mutation, you have done some work on the microorganism which you will now use for doing some new product or new processes. You don't want that culture to get into a competitor's hand or in the wrong hands, and therefore be used against your own discoveries, right? So you have to destroy that microorganism, although it's your friend in terms of trying to give you the results that you're looking at. You still have to destroy it so that your IP gets protected. So decontamination is a very important process, and steam sterilization being used for decontamination is a very important application in the industry. So you will see the 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 type of sterilizer could be generally a single door or a double door autoclave of this type. In the laboratories, for example, we use a vertical autoclave for decontamination. In BSL1, BSL2 labs, or even in BSL3 labs, we use vertical autoclaves to do a decontamination cycle, or we could use a horizontal autoclave. So we are seeing that decontamination becomes an important application, and therefore that's the third application for which you will use autoclave. So autoclave can be classified saying I'm using the autoclave for component preparation, no, I'm using it for terminal sterilization, or I'm using it for decontamination. So this is another way of classifying it. Okay. Now, the third type of classification is also based on the geometry or the configuration of it. Now, when you look at the geometry or the configuration, what do I mean? There is vertical configuration which is something which looks like this. So in a vertical plane, there is always a water tank at the bottom where the electrical immersion heater is there. And then you have the bucket in which the equipment which needs to be, or the components which needs to be sterilized is kept. And then you have a lid which has got gasket and all the things with the safety valve, with the pressure valve, and with the temperature or pressure gauge, so that you can achieve this. So vertical autoclaves are used, of course, when you're talking about a modern day autoclave. For example, they look sophisticated like this. They don't look like this any longer. They are with a proper enclosure. They come with a good control system, as I said. They have an inlet for water, so you directly connect a water source here, and there will be a level controller inside the tank which will make sure that the level is never out. Because one of the important things in a vertical autoclave, just like our pressure cookers at home, you have to make sure that the water is always present there. If the water is not present in sufficient quantity, then you would have what would happen? Can anybody tell me, if there was not sufficient water inside the autoclave, what would happen in the vertical autoclave? Just take a guess. I mean, let's assume that I was having a 100-liter autoclave, and I was supposed to keep a water level of about 10 liters in that autoclave. Instead of 10 liters, I have kept only 7 liters in that autoclave. Can any of the students tell me what would happen in that situation?
Steam would not generate.
Sir,
Steam would still generate if your immersion coil is immersed there. Okay, somebody else was saying something.
sir, maybe
Hmm.
it will blast because of the high pressure.
Okay, that's a consequence of it. But what is the first thing that will happen? That because you don't have sufficient water to displace the air, you will have air inside the autoclave, right? And that would give you a pseudo condition of pressurization. And you are right that if you don't have sufficient water, then the pressure would the heater is not able to judge that, it does not have to heat, so it keeps on heating, the pressure keeps on going up. And if there was not a safety valve on the thing, then obviously the autoclave could blast. But generally you would have a safety valve which goes off and releases the pressure, and it should give you an indication that you should cut off the supply. If you yes, do not take care of that, then it will also fly off, right? So I'm sure that we have seen videos or have maybe experienced pressure cookers in home flying off also, right? Because somebody forgot to put sufficient water into that. So therefore, the water content is important. So if you look at every modern autoclave today, there is a water level indicator. Some of it is physical. In such modern day autoclaves today, there is also an automated system with an alarm. So if the water level is insufficient, it will give you a buzzer saying water level is not there. I'm not having sufficient water. Please do something. And it will have the capabilities to abort the process and stop the process so that there is no safety compromise. Okay, so that's the vertical autoclave. You will see a lot of these type of autoclaves in laboratories, both microbiology, inoculum preparation, again, as I said, for decontamination applications, widely used vertical autoclaves even today for a lot of applications. Now you have horizontal autoclaves. What does horizontal autoclave mean? That instead of the configuration in a vertical direction, it is in a horizontal direction. And under horizontal direction, you have two types of autoclaves. You have a double door autoclave, which means you load from one side and unload from the other side. Or you could have a single door autoclave where you load from the one side and the same side you can unload it. So how do you take a decision on this? Generally, when you are talking about sterile manufacturing operations, you want to unload the sterilized products into a more cleaner area. So you would load it from one side and unload it from the other side. The loading side is generally a certain classification depending on the kind of sterile manufacturing you are doing. And the unloading side is a more cleaner area. So, and normally you attach a laminar airflow unit here at the outlet of it, so the components which are coming are protected and they are not directly in touch with the room air. So there is a good amount of or a more cleaner air, a HEPA filtered air directly touching the sterilized product, thereby maintaining sterility. So if you are doing a sterile manufacturing operation, for example, for injectable products, what would generally happen is that you would have to unload. So let me show you, just give me a moment. I'll bring out a layout again because that probably will give you a better idea of no, it is not the one I'm looking at. Just trying to look at one of the layouts which I've got. Just give me 1 second, please. Okay, so I'm going to show you one of the layouts which of an injectable facility, which is just for your information. It's I'm showing it from an interest. So this is a typical injectable facility if you look at it, and this is where the filling happens, and this filling area is a grade B area. Okay, And this is in autoclave area. So what is generally there is that this will be a grade B area. This is called as a sterile corridor or a grade B corridor. And in this autoclave, what happens is that you prepare the components and load the autoclave from one side and unload it on the other side. When you unload it here, there'll be a laminar airflow unit which will be kept here. There'll be a ceiling suspended laminar flow unit in which you will unload the component. This room is also a grade B, but there is the laminar flow area. The area under the laminar flow can be claimed as a grade A area itself. So the components which are used in the process in the filling machine, which is a grade A environment, you are making sure that it is autoclaved. So this would be a grade C area, this would be a grade B area, and there'd be a lab which will render that as a grade A area. You would collect all the components in there. You would keep them in a proper trolley and then take it into this area and mount it into this equipment, which is now got a grade A environment. So generally, this is a double door. So you load it from what is called as a loading side and unload it onto a cleaner side. This is generally one grade lower as compared to this one. Sometimes they can be even the same grade. And these kind of applications are there in microbiology labs. These are applications in sterile manufacturing processes, in biopharmaceutical, even in the for example, in the fermentation facility. So you will see a double door autoclave. Always remember, loading is done at a slightly lower grade can be done, and the unloading has to be always at a higher grade or the grade equivalent to your process areas. So if this was C, this has to be C. If this was B, this has to be B. If this was A, then you have to create an A area here, and that is important. The loading has to be one grade lower than the unloading side so that you can maintain the pressure cascades and the cleanliness of the clean rooms. I think you have a session, a separate session with Mr. Sheshadri later on, or I don't know if you have already had it on the clean rooms or with Mr. Sundar. So when they talk about the classification of the area and other things, these are important aspects to remember. So double door autoclaves, load from one side, unload from the other side. Single door autoclaves, load and unload from the same side. Now, you'd ask me, why would somebody use a single door autoclave? Sometimes you try to retrofit a facility, you may not have sufficient space, so with a proper procedural control you could do single door autoclaves. Even a lot of times your decontamination autoclaves, for example, can be loaded and unloaded from the same side because you don't have a fear of you're actually decontaminating something, so there is no need for you to use a double door autoclave for such applications. So single door autoclaves will work. So horizontal autoclave can be either a double door or a single door. Now, there are two other classifications based on the geometry. You could have a rectangular chamber, which is what is shown here, or a circular chamber, which is shown here. The initial autoclaves which were there in the industry, most of them were circular in nature, meaning that they had a circular chamber in which you had to load all the components and then autoclave it. And rectangular chambers were not very popular. The reason rectangular chambers were not popular was for a very simple reason that they were in a rectangular chamber, you have a 90-degree joint, and a 90-degree joint is always difficult to clean, is also a place where microorganisms can harbor. But this was when the fabrication technologies were not so well versed. People did not understand the science of the harboring of the microorganisms so well. There was not enough study done. Most of the modern day, but what is the disadvantage of a circular chamber autoclave is that you are limited by the lowest tangent which is there, or lowest arc which is formed inside the circle, and that is the limit to which you can load it. So if you had to put a trolley inside this, and let me go back 2 slides, if you had to put a trolley inside this in a circular chamber, The trolley cannot be of different length and size, right? So you have to have a trolley which is of a uniform size to be able to handle it properly. And in that situation, your load pattern is going to be lower, because especially when you look at the place where the diameter is there, you have more space, but because of the lowest area where you have a lower width available, you are not able to utilize the complete dimension of the circle. Okay, it's just like the famous meme which is made on a pizza box, that the pizza box is a square in which a circle is fitted, and when you cut it up it is triangles, right? So something similar here also, that it's a circle in which you are trying to put a rectangular thing, so not a very efficient way of utilizing the space which is available in the chamber. Whereas in a rectangular chamber, you are relatively able to get a higher surface area for the component loading. So therefore, rectangular chambers are preferred. Of course, there are factors like, whether this joints are properly done, whether the sealing of a rectangular chamber is better. Look, circular chamber is always better to seal. So there are advantages, disadvantages. Circular chamber, you just have an O-ring, you directly close it. It's a very neat arrangement of closure. When you talk about a rectangular chamber, to get this ring correct, to get the sealing ring right, it's also something which is a technique, because if you don't get this ring right, because this is sealing the door to the chamber, then there will be leakages from the chamber. When you have leakages from the chamber, your sterilization will not be efficient. So both of these configurations, I'm not saying which is right or wrong. It is only from a load perspective, and the fact that when you choose a supplier of an autoclave, you have to be evaluating some of these aspects very, very carefully, saying that whether the vendor has sufficient experience in fabrication of a rectangular chamber and sealing those chambers properly, so that you do not have issues. When you have a very large autoclave, which is like 2000 liters, 3000 liter autoclaves, these rings become really difficult, because the whole part has to be machined. Look, when you have a smaller rectangular, also from a fabrication perspective, machining is easier. When you have a very large autoclave, machining is a difficult task, and sometimes it is difficult to get perfect lines or squares, sorry, perfect lines parallel to each other to be able to fit the rings. The gaps can be sometimes there. So it is very important to choose the right vendor, and to have a right experience of that vendor evaluated before you pick up an autoclave, so that there are no leakages, because leakage is the one of the other deterrents to your sterilization process. If you had leakages, your sterilization process will not be efficient. You could have air and leaking into the chamber, resulting into wrong pressure and temperature data, which will not give you the stability assurance the way you look at it. So very important that while both configurations are still used in the industry, circular and rectangular today is a preferred configuration in the modern day sterile manufacturing operations, because of the fact that it gives you capability to maximize the load within a given volume as compared to a circular chamber. However, it is important to make sure that the equipment is properly qualified, and that is where the standards, that is where EN 285 comes into picture, and that is my next topic which I'm going to bring it in. This is a single page document, but I'm going to spend probably about 15 minutes on this. Okay. So EN 285 is a European standard which has been published into the industry. It's been revised in 2015, and I think there is a subsequent revision which is awaited in this year or next year. This EN 285, though it's a European standard, and there is an equivalent British standard which is also there, is widely used across the industry, including the US FDA giving a cross reference to EN 285. Okay. This has 25 sections. It's a very interesting document to read if you get a subscribed copy of it. 25 sections and 7 annexes to the document. So it goes on to describe the autoclave guidelines very elaborately. It talks about the test procedures, the criteria to confirm whether the sterilizer is safe and is delivering the operating cycle, and the load configuration, how you should load the autoclave. So when we have a discussion on the cycle development and cycle validation, I'll bring in some of the points from EN 285 in that particular presentation. But for you in today's session, I just want you to be aware that EN 285 is a specification and a standard which is used in sterilizer. So when you go into the industry, when you are trying to qualify as a quality person or as a manufacturing person evaluating any of your autoclave which are supplied to the plant or is in a process of being procured, then making sure that it is compliant to EN 285, understanding the EN 285 yourselves, and making sure that the equipment has been designed as per the requirements of EN 285 is very important, because if it is, then it actually takes care of a lot of the issues that we spoke about. Okay, it is a mandate that if you are supplying a product in Europe, then your equipment should conform to European standards or equivalent European standards, and therefore a lot of equipments have to be certified for use in Europe for supplies to Europe. So a lot of equipment, when you supply to Europe, if you were an equipment supplier and supplying to Europe, you have to meet the European standards. Sometimes when you are producing medication for those countries also, a client or your customer would look at how you have taken care of the equipment itself and to what extent you are compliant to the European standards on your equipment also. It's not a mandatory requirement that you should be, because you have sufficient validation to show that the quality of the product is not being compromised, but having a European standard being followed is important. So as an industry wide criteria, EN 285 is widely used for autoclaves, irrespective of which country you are buying the equipment for, or where you are installing it, or which country you are supplying the product for. Because EN 285, if you read this, if you ever get the chance to read this guideline and go through it, it actually gives you a good insight into how a sterilizer should be performing, how it should be qualified, how it should be working. All aspects are addressed in this standard. So very good standard to read, follow, and make sure you are compliant to it. Okay. Key definitions: measuring chain, series of elements of measuring instruments or measuring systems which constitute the path of measurement signal from the input to the output. From the input to the output, sorry, there's a spelling mistake there. So measuring chain is nothing but what are the devices and systems that you're using to measure the various parameters which are used in the autoclaving process, and how that signal is getting transferred. How are you measuring it, and how are you seeing the output of that onto the control system? Risk assessment is the risk evaluation and analysis of the equipment itself, that what can fail in the equipment, what are the risks on the equipment, including the risk of implosion or the explosion that we spoke about, or having issues in terms of compromise, leakages, etc. So what are the risks which are involved in an equipment? What can fail? How does the design of the equipment take care of those probable failures? And from that perspective, what is still a risk which is available on that equipment, or is still a part of that equipment which you sometimes need to take care through procedural controls. Okay. Risk control is the measure which is already built in, part of a risk assessment process. Software validation. This is very important. Today we are using a lot of automation, so it is important that the softwares are validated. They are working fine. What does software validation mean? It's a topic which is totally different. I don't know if somebody is covering the automation and control as a subject, or I don't know if Lakshmi Madam has spoken about some of the aspect of CFR 21 Part 11 and GAMP in her quality lectures. But software validation is a very important part, that whatever software you're using, the automation system that you're using to control your equipment like an autoclave, you have to validate it that it is working fine, that there are certain measures and controls which are built into that software, which does not compromise the data which is coming out of that system, and therefore there is no data breach. If you look at some of the 483s which were issued in right from past, but more often in the last couple of years, if you look at the 483s, you would see that data integrity issues causing a warning letter to be issued by US FDA have been one of the major areas of concern. Why is that? Because a lot of systems which are not automated, you are manually re-entering the data. In such situations, the data is liable to manipulation, and therefore data integrity is a concern. The second part is that data integrity, even in your software systems, if the softwares, the instruments are not calibrated, they are not configured properly, the automation sequence or the program is not written properly, you could have data gaps, as a result of which the data is not integral, and therefore you cannot have sufficient proof and evidence that your process is a short process. So software validation is a very important aspect of any modern day sterile manufacturing equipment. It is not only applicable in the context of autoclaves, but it is applicable to all the equipments. We spoke about compounding vessels in the past. We spoke about filling machines. We talk about terminal sterilizers. We talk about washers, primary packaging washers like the vial washing machine, the depyrogenation oven, the DHS. Anywhere that any equipment that you use in manufacturing of a sterile manufacturing process, if there is automation involved in it, then software validation is a very, very important aspect. There is a whole subject or a course on what is called as computer system validation and good automation manufacturing practices, which is if I look at it by itself is a document which is about 1,500 pages. That's just the data there, and then you have to apply that. So software validation becomes very, very important today. As software extend itself, we are seeing virtual reality, we are seeing AI come into the applications of the softwares itself. The software is an evolving technology, right? So there are newer softwares available, newer automation technologies available. So validating that from a perspective that it is not leading, the usage of the software and the automation in the control system is not compromising the safety of the patient and not compromising the quality of the product, as at the same time giving sufficient data for anybody to be able to say that a product was manufactured as per the set validated process, becomes very important. So software validation by itself is a big subject, and is important from autoclave or any equipment that you use in sterile manufacturing. Okay. Now, I have picked up one section for which is on the autoclave, which says what is the minimum documentation which should form a part of a turnover package. So the first one is what is called as a URS. What is a URS? It's a user requirement specification. So as a user, I have to provide what is it that I want the autoclave to deliver for me. Okay, so I have to write what are the essential features, what is the size of the autoclave I want, what is the type of load that I'm going to put into it, how many cycles in a day of an autoclaving am I intending to run, what is the quality of steam which I'm going to give to this. Sometimes there are geographies where it is not possible to generate steam through a separate source, and therefore I have to put it in my URS that I don't have a steam. Can you provide me a steam generator as a part of your own autoclave, for example? I Okay, if you want, I can show you this URS of an autoclave could go into like a 50-page document itself, which goes on to specify material of construction, what is the features I want, what are the alarms that I want in the autoclave for my own process, from a process perspective, from a machine safety perspective. A URS is the first document which is required as a part of your qualification of an autoclave. You have to have a user requirement specification which says what is it that you want from the equipment which forms the part of a URS. Okay, and I will, once I finish talking about these documentation, I'll probably give you a sneak peek into a user requirement document. Okay, user requirement specification for an autoclave, because I just made for one of the projects that I'm handling. Functional requirement specification. Now, this is a document which is a part of what is called as a design qualification, which is submitted by the vendor after you give the URS to him and you place an order on the vendor. He is supposed to give you a functional requirement specification along with what is called as a design qualification. What does that mean? You have given him a user requirement specification. He has given you an offer. You have shaken hands and you've given him the order. Now he's supposed to give you how is he going to design the equipment so that it meets your user requirements. Okay, what are the software functions that he is going to add into it so that it meets the data integrity requirements, the CFR 21 Part 11 compliance, the requirements of GAM5, etc. So the DQ is an important document which is given by the vendor so that you can, as a supplier, as a user, be able to evaluate that the vendor is designing and manufacturing the equipment as per your URS, and this is to be signed off. Only after you sign off a DQ does the manufacturer actually start construction of the equipment. This involves the drawing of the autoclave. It involves the complete specifications translated into engineering packages and shown to a user. The next one, of course, is performance qualification, and performance qualification starts with what is called as a, or the qualification of the equipment. I will not call it as a PQ. PQ is a terminology used in 285 and covers the entire qualification process. But in a typical qualification process, the first thing that you do after the design qualification is completed is your factory acceptance test. So the guy has built the equipment, the vendor has built the equipment for you. You go to this to his factory and carry out certain tests on that equipment, including looking at whether he has constructed the equipment as per the approved PNID, whether it is as per the approved drawing of the equipment. You measure the dimensions to make sure that the dimensions are correct within a set tolerance. You carry out certain tests, including running some empty steam cycles. You check the finishes inside, outside. So a factory acceptance test is generally performed on an equipment when you purchase it, and then you give them a clearance that okay, I have tested the equipment at your factory from whatever I could test, and it is good to be shipped to my facility. So the URS, the DQ is what you will try and check during factory acceptance test, and you can't of course check everything there because you can't carry your garment loads, you can't carry your actual loads there, right? So you agree to what tests you can do at the factory, and then based on that give the clearance for the vendor to be able to do it. So generally, as a rule of thumb, it's not a necessity, it's not a guideline. The factory acceptance test would include any kind of a test which cannot be then addressed, which is sufficient for you to make sure that your equipment which is being supplied meets the user requirement specification and the DQ, and where the further action or rectification cannot be taken at your site. Once it comes to your factory, if the dimension was not okay, you can't rectify the dimension at your factory. If the gasket was not sitting properly, you can't do that at your factory. You have to get it at the factory where it is being manufactured, the equipment is being manufactured. It cannot be done at a client factory or the manufacturing site for sterile products, right? So you will do all the tests which are ensuring that your criteria of the URS and the DQ are met, apart from the fact that anything which is critical from a perspective of rectification at the vendor's factory and cannot be done at your site are definitely checked. These are generally around any finishes. So if you had specified that I want a mirror finish with a 0.38 RA as the value of the internal chamber, and if when you measured it, it came to 0.6, you cannot go and polish the chamber at your factory. It has to be done at the manufacturer's factory itself. So factory acceptance test is an important aspect. After this, the equipment is shipped to your factory, to the manufacturing site for sterile products, and it is installed there. What is installation is? It's a physical installation, the electromechanical installation where all electrical power is given, the UPS power is given, the control system is connected, all the utilities, like you're supposed to give steam, pure steam to the equipment, you're supposed to give chilled water to the equipment for rapid cooling. All these are installed, and you check that yes, it is okay, the equipment is level, it is not sitting at an angle, so that the slope is incorrect. All those checks are done, and what is called as an installation qualification. You will check whether all the components which were shipped from the factory are correctly installed, they are correctly labeled, they are correctly installed, the insulation is done for all the hot lines, etc. So that is what falls under installation qualification. Then comes an operational qualification, where you will conduct the test, where you will take some dummy loads, you will do empty sterilization cycles using your pure steam, you will check the entire control system working fine. All these tests are done during operational qualification. And then what is called as a site acceptance test and commissioning, you would take a certain biological indicators, and you would place them inside the chamber and make sure that there is proper sterilization is being achieved on a given set of agreed parameters between the vendor and yourself. So that forms a part of the SAT and commissioning. The last activity which is done is a performance qualification, where you take the actual load. So I'm going to charge in the garments, I'm going to do liquids, I'm going to do XYZ. So you decide your load patterns, you put them inside the autoclave and run performance tests, so that you are assured that sterility is meeting. And this time you put in all biological indicators at different locations, plate them out, so that you are sure that your cycle is properly working. Cycle development is done in between the OQ, SAT, and the PQ. The cycle development has to be done, and the final operation which you do is a PQ. Sometimes you can change the sequence there, but generally cycle development is done always before PQ. After the performance qualification is done, you go ahead and start using it for manufacturing. So please remember that for any equipment which you get at the site, the qualification timelines have to be taken into account, because that itself can sometimes take easily 2 to 3 months. Okay, so generally in a sterile manufacturing operation, there is also a sequence in which you can do a qualification. If your pure steam was not qualified, you cannot use it for the autoclave cycle development, for example, because your steam itself is not qualified. So your air is not qualified, then you cannot use it. So there is a sequence in which you have to also perform these qualification parts. And I'm sure that if you are covering qualification as a topic with Lakshmi madam, she would be covering the topics of what is DQ, IQ, OQ, PQ, URS, etc. there. As I promised you, let me show you a URS, typical URS of a project. Fortunately, I have it right here. Autoclave, process autoclave. Yes. Okay, so okay, so this is a URS for one of our customers, and you see that a URS itself is probably leading to some 46 pages. That's not relevant. So generally, you give an introduction, you specify to what standards your facility is going to be qualified for. So I'm going to qualify this facility for USFDA, EMA, MHRA. I'm going to be compliant to PICS. I'm going to be compliant to WHO, GMP, NVSA, CDSU India, or MHR. So when you look at all these standards, you say that the vendor is aware that this facility is being used for supply to all these regions, and therefore he would have to also take care of it. A URS cannot be an all-inclusive document. Then I'm telling them how many days in a year I'm going to operate my plant. I'm going to operate in 3 shifts, 2 shifts. What are the kind of loads I'm going to put? So these are the key requirements. How many cycles per batch I'm going to run? How many batches I'm going to run per day? What is the working volume? So I've specified the working volume here. What is the location? What is the equipment number? What is the classification in which I'm going to put? What is the kind of automation and control I want? Then I go to describe material of construction. What is it that I want? What are the kind of polymeric materials that I'm allowing to use? What is the kind of insulation I'm going to use? What is the kind of surface finish I'm looking at, interior and exterior? Uh, Okay, then I'm talking about the operating principle. What are the functions I want? I want an air evacuation, I want vacuum, I want rapid cooling, etc. What are the utilities that I'm going to make available at the site for the vendor to be able to connect to the autoclave? So I have water for injection, instrument, air sterile, so he can choose what he needs to use and specify that. What are the parameters on which I'm going to evaluate this equipment is given here. What are the different cycles that I want to run on this? So I specified Bovidic, I specified sterilization cycles. What are the different kind of alarms I want in the system? What are the different kind of parameters that I want to be able to do? So the FO value calculation has to happen automatically every cycle. And what are the kind of samples that I want to collect? All that is specified in a URS. So this URS is a pretty large document where I have also specified that because it's a pressure equipment, what is the working pressure at which I'm going to run it? What is the design pressure that I want? What is the manufacturing standards of the piping that I'm going to use, and the manufacturing standards of the equipment itself, because I'm specifying that this has to be designed as per EN 285. Somewhere I have specified that. What is the pressure test I'm going to use, etc., etc. What are the different components that will come? So I have diaphragm valves, ball valves, angle seat valves, steam traps. What is the brand that I'm preferring to use for this equipment? Also, we have specified. What are the control variables? What is the minimum sterilization temperature, maximum sterilization temperature? What are the different requirements in terms of the mechanical components which I'm going to use inside this? Okay, you can see EN 285 references coming at a different places within the document here. then this is more like the different tests that I'm going to run on a day-to-day basis. I'm going to run a leak test. I need an alarm for high pressure, low pressure. I need a manual mode override. I need an interlock. I need data logs. I need PRV station controls, etc., etc. And then I talk about what is the kind of data storage that I need for the data which is coming out of this. So I'm saying that I need it to be CFR 21 Part 11 compliant from an automation perspective, and I want 500 GB of a data to be stored within the control system so that I don't lose it. Why is it that? Because while I take a backup, I'm going to run this for multiple years, right? So I have to be able to preserve that data, because when you face an audit, auditor can ask you for a data for the from the first batch that you supplied for a given product till the batch that you've currently manufactured. Generally, there is a 7-year cycle or a 5-year cycle. I don't know the data retention policy right now, but there is a set number of years for which you have to store the data. How do you store this data? If you have sufficient capacity of data storage within the control system, you can store it there and then do a backup of that on a server, right? Then how do you enter into the system? There are 5 levels of operation login. You cannot just have all 100% access. All users cannot have. You will have maybe an operator right, or you may have a supervisory right, or a maintenance right, or an admin right. So those are also specified. Now when you go to a software, we talk about different software features inside what we need. What are the different kind of data backup requirements? What are the different kind of reporting requirements? What are the different kind of electronic record and electrical signature formats, data management requirements? All this is specified in the URS, right? Then the URS goes to also say, how am I going to interface it with my server? It goes to say, what is the documentation package I want along with the equipment? Okay, what is it that I'm going to do during qualification? What am I going to do during FAT? I specify that also as an expectation here. What are the validation and training requirements? What is the responsibility of a vendor for making sure that he trains the staff of my company, and also goes to support me during IQAQ? What is my timelines, for example? What are the site activities that I expect the vendor to perform? And then, of course, the abbreviations which are used. So this is an elaborate 46-page document which is done for one of our clients. Not necessary that all the URSs that you see in the market are going to be of this nature, but URS is a very, very important document. So writing a good URS becomes an important aspect to get the right kind of an equipment so that you don't fail in your stability assurances. Now I'm going to touch on a subject which we are going to cover in the next one, which is around F0 values for moisture sterilization, D value and the Z value, because this is your topic which is going to get covered in the next 2 sessions. So F0 value, I'm just though these definitions are known to you, is an equivalent exposure time. So it's a measurement of time at 121.1 degrees centigrade, that the actual exposure at a variable temperature, F0 value is the equivalent exposure time at 121 degrees centigrade to that of the actual exposure time at a variable temperature calculated with a temperature coefficient of destruction of 10 degrees centigrade. So to calculate the F0 value in a moisture sterilization, average the temperature of each probe during sterile hold and then average the different temperature to get one single temperature. So there will be multiple probes which will be kept inside the autoclave, and that will be averaged to get one single value. And to the actual exposure time, you will say what is my equivalent exposure at 121 degrees centigrade. And that measure of time is what is F0 value. Right. The D value is the time required to change the reduce the microbial population by 1 log reduction by 90%. Okay. The formulas for F0 is given here. I'm going to elaborate that in the next session. And delta T is an equation in the equation is called as the lethality rate. So lethality rate is important because if you switch the temperature, you remember the sterilization decision tree process. We said by changing the conditions, you can still achieve moist state sterilization. So it is that lethality rate. If you are able to calculate from F0 and D values, you can say whether changing the conditions will still give you a sterility assurance. So this is the next session. We are going to dwell on F0, D, Z values and how they are calculated. Those will be the principles which will be there. We will discuss more about it. Now I'm going to leave the open for question and answer on the types of autoclaves, including the topics that we covered yesterday. So please ask any questions that you may have on the types of autoclaves itself. Any questions, students? Students can ask questions.
Sir, is there any website to see how it works, autoclaves and sterilization process?
You can find certain videos on YouTube on the principle of working of an autoclave.
Okay, sir.
But I don't know how good or bad they are because I haven't attempted to see them. For me, the best way of visualizing this is just visualize your own pressure cookers at home to see how sterilization occurs there. So, yes, I know it's a crude way of looking at it, but if you look at it, you put in water, you put in the component there, you close the lid, you allow the gas to generate the steam. When you generate the steam, initially you will see hot air coming out from the top, right? And then you see a continuous steam or stream coming out, and then you put the safety valve on top of it so that you are able to then start the whole time. And then you see that when the if the pressure goes above a certain value, in those cases there's a preset pressure. But in your case, in our case in manufacturing, we are going to control the pressure. In that case, it's a preset value. The safety valve goes up and down to release the pressure, and then after a certain period of exposure time, you close it. If you are a microbiologist and if you were to run a pressure cooker properly and take that sample and plate it, before and after, you will find a reduction in microbial count. I don't know whether it will be 10 to the power of 6 log, but it's a good example of visualizing things. But check YouTube. There will be some videos which may show you that. I haven't checked any YouTube videos for uh, working principle of a thing. There are a lot of videos on terminal sterilization for sure, which I don't know if I showed you in the last session when I covered terminal sterilization, but autoclave I'm not too sure, man.
Okay, sir.
Okay, any other questions? Okay. If there are no questions, let us end the session here. Let's meet exactly in about 15 minutes at 10:30, where I will take up the principles of sterilization in terms of FO, Z, and D values, the working principles of an autoclave, and let us have a discussion there. So 10:30, it is about 10:17 now. So 13 minutes, we again get together and start our discussions. Is that okay?
Yes, sir.
Okay.
Okay, I'll see you in another 15 minutes.
Thank you, sir.
Thank you. Okay. So please let me know once everybody is back so I can start the session.
Okay, sir.
Okay, shall we start? This is okay. This is a series, lecture two lecture parts, which we are going to talk about autoclave principles and operations. I'm not going to dwell on the working operations and principles because we've already covered it in the previous session, but a couple of slides just to recap on what we've said. We are going to concentrate on this session on what is D value, Z value, F0 value, and what is the importance of that, and how do you compute these things. So one of the things that we are going to do in this session is I'm going to give you uh, some statements based on which I expect that students will just work out the D value or the Z value or F0 value. at instantaneously and come back to me with what their results are. It's not a test of a thing, but just to make sure that you've understood the principle. So we'll do some exercise to keep a pen and paper or a paper and a pencil ready as we progress on this lectures. Okay. We're still talking about the moist heat sterilization principles. And that's where we are going to talk about it. We have seen the different phases of sterilization. I'm not again going to go through it. These are the 6 factors which are critical for a successful steam sterilization: time, temperature, moisture, direct steam contact, air removal, and drying, which is important. So we are now going to get into what is an F value, F0 value, what is a D value, what is a Z value, how do we compute them, and what is the importance of that. So F0 value, it is referred to as F0. If it is not FO, it is F0. F0 value is the equivalent exposure time at 121.1 degrees centigrade to that of actual exposure time at a variable temperature, calculated with a temperature coefficient of destruction at 10 degrees centigrade. So this is just a definition. I will come to the detail of this as we progress on the thing. I just want you to remember these definitions. To calculate the F0 value in a moist heat sterilization, average temperature of each probe is taken. We saw this in the last session during the sterile hold, and then the average of those temperatures is taken to one single temperature to compute the F0 value. Now, D value is the time required at a specified temperature to reduce the microbial population by 1 log reduction, or 100% to 10% or 90% reduction. Z value is the number of degrees of temperature required to be increased, which will affect a tenfold variation in D value. So since D value is at a specified temperature, if you increase the temperature by a certain number of degrees, then a further tenfold variation will happen in a D value, is what the Z value is referred to as. F0 is calculated by using the formula delta T multiplied by 10 to the power of T minus 121.2 divided by Z, where delta T is the time interval between the two temperature readings. Okay. The T is the actual temperature at which the product is under sterilization. So 121 is the ideal condition of saturated steam at 1.2 bar. But if your actual temperature was happening at, let's say, 125 degrees centigrade, and we saw other sterilization temperatures like 132 degrees centigrade being used, or sometimes you would use a lower temperature for 115 degrees centigrade in terminal sterilization, for example. In such situation, when you take the value here and divide it by a temperature coefficient, which is assumed as 10 degrees centigrade for the formula, but there is a scientific way of calculating that also, you will arrive at the F0 value. And once you have the F0 value, then you say, okay, this is the time for which I'm going to expose the product so that I achieve my sterility assurance. So these are the formulas. Now, before I get into the calculations part, let's just understand the thermal day threat curve, or the decimal reduction time curve, or the survival curve. So the D value is nothing but decimal reduction that we are talking about. And as we said, it is 1 log reduction that we are talking. So that is a D value in terms of time. It is always measured in time. Okay. It is at a specified temperature. So you say my D value at 121 degrees centigrade is so much. Okay. Is say 10 minutes or 15 minutes, whatever it is. So when you use the notation D, you should always accompany it with the temperature. So you see, I've written D at 121 degrees centigrade. This is the curve that I've got. So D at 121 degrees centigrade. For this, if I had a number of living bacteria as 1 million, and I was trying to get it down to say 10 as a final count, then my D value to take a reduction from 10,000 to 1,000, which is a 1 log reduction, is going to be about from where starting here to here. So that's about 15 minutes. And this is generally a linear curve. which you see. So it's a linear curve that you see plotted in terms of the time and the number of living organisms, or a logarithmic scale. There is another way of expressing the same value or the d value, which is by looking at the number of survivors. So this was the number of living bacteria which was there, or the number of survivors, the same thing. But the you take different spores. So you say vegetative cells, you say C. bolotinium spores, you say another thermophilic compound like say Geobacillus stereophilus, and then you say what is the time that is taken, and then when you see a one log reduction, you get the time from a linear curve. So you could measure the number of survivors at a given temperature, plot it at different logs that you get. So you take samples at different time, plot it, take the linear curve, and then see where you have achieved a tenfold reduction by plotting various points, and that gives you a d value. So the thermal, that red curve also can be plotted. Higher the load, the inside the chamber, longer is your time for achieving this one log reduction. So it is important that when we do a cycle development, when we do a loading of an autoclave, we optimize the load so that you are not taking too longer time in your manufacturing process. So is this concept clear of what is d value? Because this is important, that at a given temperature, when you achieve one log reduction or a 90% reduction in the bacterial load, that is called as a d value, and it is expressed as a always in time. Okay, clear? Now let's look at the d value for some of the cultures that we have been using in the industry. And I've gotten some cultures here from a perspective of food industry also, because I have used some examples of food industry in the calculations. So Bacillus stereothermophilus takes about 3 to 4 minutes to achieve that kill rate or one log reduction. Bacillus subtilis, which is also something that we will use for some of the applications, takes about 0.3 to 0.7 minutes. Bacillus coagulans is used, takes about 0.4 to 0.8 minutes. Clostridium sporogenous takes 0.4 to 0.8 minutes again. Clostridium thermosaccharolyticum, sorry for if my pronunciation is wrong, 3 to 4 minutes again. Clostridium botulinum type A and B takes 0.1 to 0.5, and then there is D-sulfotomaculum nigrificans, which takes 2 to 3 minutes. So you see for different species, the D value is different at 121 degrees centigrade. And that is what you need to study as microbiologist, if there are microbiologists in the room, to say that if I had a particular flora in my environment or in my load, and if I want to kill it, what is the d value of that organism? First of all, I should know. If I know the d value of the organism, then I can say my initial load is so much, my final load is a reduction criteria of 10 to the power of 6. And if I want to do that, then this is the minimum cycle time I need to run theoretically. Then you actually do a practical test for that and make sure that you are achieving the reduction, and by putting biological indicators there, by using a more tougher spore, and then you see, okay, now this is the cycle that I'm going to run, that I will need 12 minutes to do the achievement, or I'll need 30 minutes to do that. That is the importance of d value, that you should first of all know what organism you're dealing with, and knowing the d value of that organism helps you in arriving at the time cycle that you will look at. So if you had a flora, generally my microbiologist will come up to me and say, I don't have just one organism, I have multiple organisms there. Choose the toughest of those organisms as your criteria for arriving at your thermal death curves. And if you don't have anything else and everything is, say, like you had Bacillus subtilis, Bacillus coagulans in your flora, and then you were not too sure what to do, then use Bacillus stereothermophilus as your criteria, and then develop your cycle based on that. Because anyway, that's your biological indicator, which you'll use as per USP to validate your cycles also. So from that perspective, knowing the organism becomes very important. So generally, when you have a sterility failure, you will do a profiling of the microbial organisms which are present in the environment or present in the loads, and characterize them to find out what is the microorganism, so what family and what sub family they belong to, and then based on that you will arrive at a criteria of how to sterilize them. Okay, so very important information that will be used by microbiologists when they are defining a certain sterilization cycle or recommending a certain sterilization cycle. Now, thermal curve or the thermal death rate curve is of importance to us only if we are able to take samples and then plot it and say I received one this thing. So that exercise is of course important. But generally, what you will do, you will start at a particular load, end at a particular load. Now you will have to arrive at a what is called as a thermal death time curve. It is the time in minutes required to inactivate an arbitrary chosen number of spores of a given bacteria at a specified time. What does that mean? That means that you will start with a certain load, you will give it a certain time for it to get killed, and then you will plot that to see that what if my spores were, let's say, 10 to the power of 4, and if I want to go to the 10 to the power of -1, and if my temperature was at a certain position, what is the reduction that I'm going to receive? So it's a logarithmic death time plotted against the corresponding temperatures. And when you do that, you will get a curve or a linear graph again, which will tell you that what should be that temperature. So remember, what was the Z value? It is the number of degrees of a temperature required to increase the D value by 10. So you will calculate the Z value also. Why do you need to calculate Z value? In your F0, you need to put in a Z value. So a Z value is sometimes chosen as 10 degrees if the organism is known. But how do you arrive at the 10 degrees? It is based on thermal death time curve. So you will plot the D curve and you will plot the thermal death line curve for a given microorganism to say what is my D value and a Z value. All right. And therefore, you can arrive at your F's value. So Z value is the number of degrees of temperature required to increase, which will affect a tenfold variation in D value. The slope of the TDD curve is defined as Z. So if you take a slope of this curve, you know how to measure a slope of a curve, then it will tell you that if I have to traverse 1 log, this is the temperature difference I need to make. So the Z value for Clostridium botulinum is 10 degrees centigrade, while for Bacillus subtilis, it is 6.5 degrees centigrade. So if you change the temperature or increase the temperature by 10 degrees centigrade, Clostridium botulinum, you will see a 1 log reduction there. If you had Bacillus subtilis and you change the temperature by 6.5 degrees centigrade, you will achieve that 1 log reduction. So Z value is important because you want to know what temperature difference you have to bring in. Okay. Now F value. So I'm going to start with what is called as F value and then tell you what is F0 value. Okay. So the F value is the sterilizing value in minutes, time in minutes, required to kill an organism in a specific medium at a specified temperature. So F value is a general definition here. Why am I giving you this definition? Because you will use F value in different applications. For dry heat sterilizer, you use what is called as FH value. In case of autoclave, you use F0 value. So F value can be applied to any sterilization technique, which is using thermal sterilization technique. Okay. where it is the time which you measure in minutes required to kill an organism in a specific medium. Why is it specific medium? Because you have a particular media in which that organism is exposed. You are trying to kill it in that specific media, and for that specific media, there will be a particular time. If the media is changed, then the time will also change. So you have to define the specific media. You have to define the time in minutes, and at a specified temperature, the time required to kill an organism is called as an F value. Now, F0 value is a time equivalent at 121.1 degree, 121 when you measure the actual exposure time. So when you specify the temperature is 121 degrees centigrade, then and measure the actual exposure time, that is where you call it as an F0 value. So F0 value always is at 121 degrees centigrade as a baseline temperature. Understood? Now, to calculate the F0 value, we have seen what is the formula. Now, we are going to do some examples of how to calculate this F0 values here. But before I get there, I want to show you what is a 12D concept, or it may make you explain what is a 12D concept. It is a method of expressing the process lethality requirement. So when I say I want to achieve a certain reduction, I'll specify whether I want a 10 to the power of 6 log reduction, whether I want a 10 to the power of 3 log reduction, whether I want a 10 to the power of 9 log reduction, and so on and so forth. Now, what is generally done is that if you want to go to a stage where you want to do an overkill approach, meaning that you want to reduce, you want to go ahead and kill the microorganisms for sure, and you don't want to take a chance that I will go to a 10 to the power of 6, but I don't know whether in that media you will still see a growth, because I'm going to keep the product for say 2 years. It has got a stability for 2 years. So at the end of 2 years, what may happen? I want to go with an overkill approach so nobody is dead. Then you use a 12D concept. It's not a very efficient way of doing it, because what happens is that if you go to that overkill approach, then you will end up having a lot of lot of wastage of energy in trying to achieve that. So if you know the load is never that high, then don't go for a 12D concept. Otherwise, if you don't know, if you are playing in a blind, then yes, a 12D or overkill approach always works. So one of the widely used practices for steam sterilization efficiency is that you take Bacillus stereothermophilus and see whether 10 to the power of 12 reduction has occurred. And then you are sure that your process is working fine. But if you were to profile the bacterial spores, and if you knew that this was the bacterial load which I have, you don't need to go for a 10 to the power of 12 approach. The 10 to the power of 12 approach is used when when you know that there are toxins present, a lot of toxins present. For example, when you are using, let's say, a media where water is used, and you want to be sure that the water is sterile because you are then using it for growth of microorganisms, of inoculum, etc., then yes, in that situation you can go with a 12D approach. So there are applications where you'll go with a 12D approach and applications where you'll not. But if you were to do a regular load of garments, etc., then 10 to the power of 6 generally is more than sufficient for us to achieve. Okay. This is also a concept which is widely used in the food industry, because the food has to be packaged sterile. All the food that we get is ready to eat today is sterilized, is autoclaved and given. So in that situation, a 12D approach works because there are large batches. Okay. So 12D concept is a approach where you reduce the load of Bacillus stereothermophilus by a factor of 10 to the power of 12. And that's a overkill approach. So we did talk about overkill approach in one of the other sessions that we had. I'm now going to talk about certain formulas, and these formulas is what we are going to use is as examples for doing out some examples, and then I'm going to give you certain problem solvings which you can do and come back to me with a with a certain um, uh, answers, and let me see how well you've understood this concept. Because on the session as such, I only have to tell you what are the formulas, how to use the formulas, and what are the data points that you need, and you can arrive at yourself by using these values. So let us look at it. The d value is the time to required to kill 90% of the population in a definite time in a definite media. So first and foremost, you should know what media you're talking about and what timelines you're talking about. To determine the d value, survival plot is obtained by plotting the log of microbes against a particular temperature. For a survival plot, it is observed that a particular temperature, the number of microbes are decreased by a factor of 10. So you have to first of all know what is the media you're using. So let's say you use Bacillus stereothermophilus. It is in a media, you know a number of spores that start. You will subject it to a particular temperature for a definitive time and start plotting it. So you will say, do it for 10 minutes, take the plate out. Do it for 20 minutes, take the plate out. Do it for 30 minutes, take the plate out. And you will see what is this number of surviving spores at the end of 10 minutes, 20 minutes, 30 minutes, 40 minutes, 50 minutes, and you will plot it for stereothermophilus, Bacillus stereothermophilus. Once you do that, you get the curve. Now, when you get a curve, you know what was the time taken for a 10 log reduction through multiple data points. So you will see initially my in 10 minutes I achieved. So when you plot that and plot it on a logarithmic scale, and you see where the 1 log reduction occurred, you see the time. You say, okay, it took 10 minutes here, it took maybe 11 minutes here, you maybe somewhere it was 10.5. So you then say, okay, fine, my time which is working in this media is, let's say, 11 minutes for that log reduction. So I take that time, I take the star N0. N0 is the initial load and the load at the end of the time, and that is how I arrive at a D value. So it will then give me the time which is required at a definitive temperature to go from value X to value Y. So I started with 10 to the power of 6 spores. I went down to 10 to the power of 3 spores at a particular temperature. What was the time taken? That is what is your D value. Now the Z value is the corollary or associate of D value, where it says if I change the temperature by so many degrees, I will get a 1 log reduction, right? So what will I do? I will take temperature 1, temperature 2. I will plot it against the log value of D1 and D2. So I will say that my D1 was say X, my D2 is Y, and my temperature 1 was say 132 degrees centigrade, temperature 1 was 121 degrees centigrade. By making that change, what is it that I get? So this is what the formula for Z value is. The procedure to find the D value is the desired microbial is inoculated in a particular media and incubated for 24 hours. The initial microbial population is measured using a spectrophotometer. The incubated culture is exposed to heat at 65 degrees centigrade. Final microbial population is measured at using a spectrophotometer. The initial and final microbial population is put in the D value formula to arrive at the D value itself. So this is a laboratory method where you don't actually expose it to high temperatures, but you just do it at 65 degrees centigrade. You can do that at 121 degrees centigrade. You can do that at any other temperature, but you have to base it on a media and a temperature. So it's media, temperature put together, which will give you. And the media should be such that it does not get charred or it does not get burnt out when you expose it at that temperature, because that will give you a wrong reading in a spectrophotometer. Why a spectrophotometer? Because you are using the UV value there in a spectrophotometer to count the number of population and to arrive at the total count there. Otherwise, it is very difficult to count 10 to the power of 6, 10 to the power of 3 physically. You have to allow them to sporulate to that level, right? So you use a spectrophotometer for that. So this is the procedure which is defined in the literature, saying this is how you do a D value. Procedure to find a Z value, you repeat the above procedure at a different temperature, say 75 degrees centigrade, and then when you put the value of the D for at 75 degrees centigrade and 65 degrees centigrade, you will get a Z value. And you know the temperature is 10 degrees centigrade in this case. So uh, looking at this is an exercise which has to be done where you know a given microorganism, you arrive at a survival plot, you arrive at a Z value at different temperatures and publish this data. Fortunately for the microbiologists today and for the industry, this data is already available for a lot of microorganisms which are characterized and found in the industry, and therefore that data is available. Only if you encounter a completely new microflora where the family or the subfamily is not known, then you get into an issue of having to carry out this. And that is the reason why I thought it was important for us to understand how this exercise is done, so that in case any one of you who is in the microbiology section of a company or a microbiologist need to arrive at a D value or a Z value, then you know how it is done as a actual practice in the laboratories. What is the difference and similarity in D value and Z value? D value is not necessarily dependent on a temperature, because you are fixing the temperature and then arriving at the log reduction. In a Z value, you need to know what is the delta of temperature that you have gone so that you arrive at a Z value. D value is defined at a particular temperature and it is dependent on time. It is measured in minutes. Z value is you are varying the temperature. It is, and then you are measuring that in Celsius. So Z value is always expressed in degrees Celsius. D value is always expressed in minutes. Remember this as a major difference. The similarity, both are used in the area of sterilization to assess the efficiency of the heat to kill the microbes. It tests the sensitivity and the heat resistance of microbes towards the heat. Why are these important? Why are they used? They are both associates to each other, because while in one case you have a defined temperature, you know by varying the temperature what is it that you can achieve in terms of the kill rate, and therefore you can vary the parameters of your sterilization to achieve a sterilization and to make a decision if moist heat sterilization can be used. So go back to your sterilization decision tree. If you were to change the condition, if you knew the D value, and by playing around with the Z value, you could at a given temperature or a given higher temperature be able to achieve a moist heat sterilization and therefore avoid getting into other requirements for aseptic processing in this situation, right? So this is uh, this is what is very important for us to understand the variation between D and Z value. Okay. Now, okay, these are calculation examples, so I'm not going to get into that right now in this session. Before I get into this, I would like to just take a short closure here to say whether all of you have understood the concept of F value, D value, and Z value well. If not, let us discuss and debate this a little bit, because it is important that all of you understand these concepts pretty well, because you are going to use this in the industry again and again. So I would like to see questions or clarifications any one of you might have on DZ value and F value, if you have at this stage itself, before I take you through the calculations.
So,
Yes.
so after finding the D value and Z value, we will go for F0, sir, for calculation.
You can go with an F0 value. Yes, you can go ahead and find out what is the F0 value at equivalent value at a given for to achieve a equivalent time. The F0 value is calculated having the Z value as a known factor. If you don't know the Z value, you can't calculate the F0 value. You can assume a value of Z, but then what would happen is you will either get into an overkill or an underkill.
Okay, sir.
Any other questions?
Sir,
Yes.
can you explain the Z value again? Sir, how to plot the graph of Z value?
Okay. So what you will do for determining the Z value is that what is Z value? Z value is the change in the temperature which will make the D value change by 1 log reduction, right? So what does that mean? What is D value itself? It is the time required to change the population from say 10 to the power of 6 to 10 to the power of 5, right? So when we go to a D curve, what does it mean? You had 10,000 spores, you went 1 log reduction, which is 1,000 spores. The time taken was so much, right? Now, when you want to do a Z value, you are saying, okay, this plotting was done at what temperature? 121. If I now change the temperature from 121 to 132 degrees centigrade and I do the same plotting, what is this change that I'm going to see? When, if I do that 1 log reduction, if I go from 10,000 to 1,000 at 132, what will be that change? So the time that I will get here would be different as a D value when I change the temperature, correct? Yes or no?
Yes, sir.
Okay, so so by changing the temperature, I'm going to be able to determine what where do I get that addition 1 log reduction by change of temperature. So I was at 235 degree Fahrenheit or let's say 121 degree centigrade, and I changed the temperature to 130 131, and wherever I get this 1 log reduction on the lethality or the surviving organism, I know that by changing the temperature by 10 degrees centigrade, I am able to get a 1 log reduction quickly. So what will I do while doing the Z value determination is, I will first find the D value at 65 degrees centigrade. Okay. Then I will find the D value at 75 degrees centigrade. This is for this is taking into account the some of the cultures which get sterilized at 65 degrees centigrade and are found generally in the food industry, for example. But if we are talking about sterile manufacturing, then we use 121. So I do, let's say, Bacillus stereothermophilus. I plot the graph at 121 degrees centigrade. I plot a graph at 115 degrees centigrade. I plot a graph of D for determining the D value at 132 degrees centigrade. Now, for me to determine the Z value, I will take the D value where I got a reduction of 10 log, 1 log at temperature 121 degrees centigrade and at 115 degrees centigrade, and see whether by getting that log reduction, am I able to achieve a same kill rate that I am trying to achieve, and therefore reduce my time. Because what is the D expressed is? D is expressed in time. By determining the Z value, if I change the temperature by 10 degrees centigrade, if my time is changing from say 15 minutes to 10 minutes, I will be able to achieve the same kill of 1 log in 10 minutes against 15 minutes by just changing one factor called temperature. So I'm trying to establish a temperature for a given time at which I will still achieve the same log reduction that I achieved at a different temperature. That is my Z value. Okay.
Okay, sir. Thank you, sir.
Okay. Any other questions? Okay. So what I'm going to do is I'm going to show you two examples here of how an F0 value is calculated. And I'm just going to leave this as examples for you. And then I will stop the session and give you a short break. When we come back on the next session, I'm going to give you some data points and you will have to calculate the F0 value and tell me what your answers are. Okay. So I'm going to take organism Bacillus stereothermophilus, a friend we know. We like this guy because we use him on a biological indicator for taking our sterility assurance levels and getting a sterility efficiency determination. So, for example, if D121 is 1.5 minutes for Bacillus stereothermophilus, so D is 121, which means in 1.5 minutes, my reduction is happening from 10 to the power of 1 to 10 to the power of 0, or 10 to the power of 2 to 10 to the power of 1. Okay. If my initial load is 10 to the power of 6 and I'm looking at a final value of 1, which means, or minus 1, which means that I'm looking at a reduction of 6 logs. All right. Then what is my F0 value? I will take 1.5 minutes will give me 1 log reduction, I know. For a 6 log reduction, I will multiply that 1.5 by 6. So in 9 minutes, I can achieve a 6 log reduction at 121 degrees centigrade for Bacillus stereothermophilus is what I calculate. So my F0 is a expression in minutes at 121 degrees centigrade, by which I will receive a 6 log reduction. So I can determine what is my sterility assurance requirement. For example, I say I want my 12 log reduction. So my log 12 minus log 1 is what I will plot. And I will say that I will need 12 log reduction. My D121 was say 1.5 minutes. So 1.5 into 12, 18 minutes is the time I will require to run my process so that I can achieve a 12 log reduction. In this example, where I have taken D121 as 1.5 minutes. If D121 was a different value and I give you two different values of the N0 and NT, then accordingly your time will vary. So why is this F0 important? Because when you determine why and how will this be used in your autoclave cycles, is that you will take the you will determine it based on Bacillus stereothermophilus, because that's the most heat resistant spore which is known to us. And you don't know what are the different kind of microflora which is there, because and you're not even you might have profiled the micro profile of your factory and you might not have found anything more tougher than Bacillus stereothermophilus. So you will use Bacillus stereothermophilus as your sterility assurance factor on a biological indicator. So what will you do is that when you put it on a given load, okay, you know that if I had a bacterial biological indicator with 10 to the power of 12 spores already on that indicator. So the indicators are actually spiked indicators where they have a certain quantum of Bacillus stereothermophilus on that strip. Okay, so you might have 10 to the power of 12, 10 to the power of 6, 10 to the power of 9. You get different type of strips. You take these strips, you put them inside the autoclave along with the load which is there. Why do you keep it along with the load? Because the load determines the pattern in which your steam will flow and sterilize, will reach every corner and do the kill. So you will take this stereothermophilus biological indicators, place them in various locations in the autoclave, and then you will subject it to 121 degrees centigrade for a certain time. You know that if I want to reduce it by 10 to the power of 12, I need to run a minimum cycle time of 18 minutes. In this case, let's say that the biological indicator that you got had this data that my D121 is at 1.5 minutes, and it is 10 to the power of 12 biological indicator that you've got. When you calculate the F0 value, your answer will be 18 minutes. So if you were to run the sterilization cycle for 18 minutes, then you should see a 10 to the power of 12 reduction. So you keep all these bacterial spores strips inside at strategic locations inside the chamber along with your load. Now you run your cycle for 18 minutes. At the end of 18 minutes, cooling down the autoclave, remove those bacterial strips, your biological indicators, put them into the test media and see whether your spores or the spectrophotometer and count the number of spores. If the spores have reduced from 10 to the power of 12 to 0 or 1, it means that your 18-minute cycle was okay. But if that had not gone and you saw 10 to the power of 12 going down only to 10 to the power of 9 or 10 to the power of 6, for example, you have achieved only 6 log reduction. Okay. Your aim was to achieve a 12 log reduction, I'm saying. Now, in that situation, what does that imply? That your 18 minutes is not sufficient because of the conditions inside the autoclave. So you will first of all find out whether your air, there was any air entrapment, whether there was any load which did not allow the steam to go through properly in a chamber. So let me just go back to the previous presentation that we ran, so that I can use the autoclave profile and show to you what happens there. Okay, let's use this as the picture. So this is an autoclave load which is there inside. So you will place your indicators maybe on the tray here, on the other end of this tray, maybe some tray in between, maybe at the bottom tray. There are some locations which you will determine. You will place it on the validation port is here. So there's one opening here. You will place it near the drain. Everywhere you will place those spores. Now your steam is flowing from the top. It is trying to sterilize the whole load and going out. You will then collect those strips, knowing the location, and say whether I achieved the kill rate which I was aiming for or not. I ran the cycle for 18 minutes. Ideally, it should have gone to a 0 kill, but for some reason it has not gone. Then I need to look at how I have placed my loads. Is it too tight? Is it touching each other? As a result of which steam is not able to penetrate. Because remember that in moist state sterilization, what is important? What is critical? My steam has to penetrate. My moisture has to penetrate the load. If I don't do that, then I will not achieve the kill. So as a result of which, the bacterial indicator gives you that by taking the Z value, by determining the F0 value, you first define the cycle time. And after having defined the cycle time, now you are getting into seeing that it is not sufficient because the way I kept the load. So you will investigate, you will either rearrange the loads and rerun the cycle, or you will increase the time, which is where Z comes into picture. And you will say, okay, I was running it at 121 degrees centigrade. Now I have two options. I can increase the time. If I increase the time from say 18 minutes to say 25 minutes, am I achieving a reduction in time? If I am achieving the load reduction of sterility assurance of 12 by increasing the time from 18 minutes to 25 minutes without changing the temperature from 121 degrees centigrade, I will fix that as an F0 value for this load in the autoclave. I can also do another thing, which is change the temperature, right? So I can say that if I had 132 degrees centigrade, this D value would have been lower, could have been 0.5, could have been 0.75. So I should have achieved the reduction in say, again, maybe 9 minutes or 6 minutes. But now I am required to run the cycle for a long. So F0 is a determination which will say how long you have to run the cycle to achieve the required sterility assurance level for a given organism species, Bacillus thuringiensis being used as a standard organism if no other known tougher organism is available. Okay, so F0 calculation is done for every batch on an autoclave to make sure that your loads were running consistently. You cannot have one cycle. You had a load of 10 garments, 20 spatulas, 15 filling nozzles kept in a load. And one cycle you ran it and you achieved, you had a validated cycle which said you should ideally run this cycle for 20 minutes of sterilization whole time, which here we are talking about the whole time. Okay. So I was supposed to run it for 20 minutes. You ran cycle number one, your F0, the calculation which was computed there came out to be 20 minutes. In the second cycle, it took you 25 minutes. Third cycle, it took you 15 minutes. Is that a deviation from the validation? Yes, it is a deviation from the validation. Then you have to investigate and say what changed between those 3 cycles of autoclave that I got a time which was different from what was validated time. For a given load, you establish an F0 based on Bacillus thuringiensis, and that has to be repeatedly be achieved every time. If you cannot achieve that, if your Bacillus thuringiensis biological indicator, which you kept with every autoclave cycle, was a different data point, then there is a problem in the autoclave which you need to address. Maybe the steam flow was not proper, or whether the air evacuation was not proper. So this is an important factor which gives you the performance of a autoclave. Okay. Now I'm going to use another example of Clostridium botulinum. It's a commonly found contaminant in the food industry. The expected log reduction, there is a 12 log reduction because there you don't want to take chances because food has a very low shelf life. They are generally kept, look, in pharmaceuticals, you have storage conditions which are well defined. In case of food, the food could be moving through a logistic chain where the temperatures can vary from room temperature or cold temperatures to high temperatures and back to cold temperatures, right? So in such situation, the best method of approach is to go for a 12 log reduction or a overkill approach. So the temperature sterilization is 250-degree Fahrenheit, which is used in the food industry. The value of this organism at the temperature of 250-degree Fahrenheit is 0.21 minutes. So now if you want to achieve a 12 log reduction, 0.21 into 12, 2.52 minutes is your F0 value. If you were to keep it in a moist sterilization situation of autoclave. So in 2.52 minutes, you should achieve a 12 log reduction. If you don't achieve it in 2.52 minutes, you'll have an issue. So why is this important in a food industry? Because in a food industry, you don't have a luxury of a batch autoclaving. In a sterile manufacturing facility, you have the possibility that you will do the autoclaving under batch operation, meaning that you load the autoclave, you finish your autoclaving cycle, unload it on the other side, prepare the second load, load it after that, and run maybe 5 or 6 loads in a day. In a food industry situation, as if as microbiologists you ever went into a food industry, then you would see that the food manufacturing process is continuous manufacturing process. When you see a continuous manufacturing process, these things are running into autoclaves of a different kind where it is continuously exposed to a certain tunnel kind of an autoclave which is there, where it goes into an autoclave and comes out. So it gets heated up, gets exposed, comes out on the other end, and you have to achieve that 12 log reduction. That residence time for it to stay inside the autoclave before it gets unloaded is determined by F0 value. And if you don't have this data with you, you won't be able to determine what is the right time of exposure at that temperature, and therefore you will not be able to achieve the right kind of a reduction. You could have a contaminated food at the end of the day. can be applied in principle for moist heat in a closed condition. So how does it work on a batch operation? You load it, you unload it in 2.52 minutes. This is quick because in 2.52 minutes is not a very long cycle. So you do it for 2.52 minutes or 3 minutes, take it out on the other side, load the next lot. Come, so every 2 minutes you are taking out certain quantum of food from that autoclave, and it is already autoclaved, and then it's fit for distribution. So these are measures. This is how you will use F0 and D values in your practical experience. So with this, I'm going to end this first session on F0, D, and Z values. When we come back, I'm going to give you some examples to calculate D value, Z value, F0 values. And I would expect all of you to have a pen and paper ready and do that calculation, and maybe present your screens and show me how you've done the calculation. Okay, so that it becomes a very important topic, because except for showing the formulas, I don't have anything else to talk about much here, and the importance of these three factors. So let's do a few exercises so you get familiar with how to do calculations. But before I end, if there are any questions on whatever we have discussed so far, even after you looked at the examples, please ask the question. Otherwise, I will we will join back in about another 10 minutes or 12 minutes, maybe 10 minutes, yeah, 11:35. And then we will quickly go through a few calculations so that I'm clear that all of you have understood the concept clearly. And then we can end the session for today. Okay. So any questions before we take a break? No. Okay, fine. So let's take a break. It is 11:26. Can I request everybody to be back by 11:36, please?
Yes, sir.
Okay. So I'll see you at 11:36. Units for Bacillus stereothermophilus. The N0 value is 10 to the power of 12, and the expected NT at is 10 to the power of 3. Please calculate the F0 value. So just work on this equation right now. Work on this problem right now. Once you finish, let me know that you've finished, and I will pick up a student who can then present their work and see how it was done. Okay. I'll also give you the second problem right away, so you can work. Okay, let's do one problem at a time. So first work on the first problem itself. Everybody has noted down the figures. Yeah. So please work on it. And once you're done,
Sir,
sorry.
can you share for one more time?
Yeah, sure, not a problem. D132 degrees centigrade is 2 minutes. N0 is 10 to the power of 12. NT is 10 to the power of 3. Please calculate the F0 value. Okay, are you ready? Any of the students were ready with the F0 value? Hello? You should not be taking so much time. It's a simple calculation.
Sir, I got as 18 minutes, sir.
Okay, can you show me the computation you've done? Oh, okay, you've done it on pen and paper, right? So, okay.
Yes.
So tell me what is the formula you used and how did you arrive at 18 minutes?
So I use the formula that t is equals to dt log no minus log nt,
Hmm.
and whereas mm, for
D is
the
what?
d is 2 minutes, sir.
Okay.
That was the microorganisms,
Yeah, okay.
and mm, it was
And N0
yeah,
is?
n0 is the initial reduction, that is initial it was log 13, and
Log, log
after
13. Log 13 or
log log 12, sorry sir, log
okay.
12,
Hmm.
and final it is reduced to log 1, log 3, sir.
Hmm.
So if we calculate, sir, we get a log 9, sir.
Hmm.
And if we multiply with the d value, we get the 18 minutes, sir.
Okay. Good. Correct. That's correct. So your answer is correct. That is what it is. Okay. Now I'm going to give you the second problem, and sorry, who? So Joshua is your gave me the answer. So the next problem, Joshua will not be the person who will answer the question. I'll want somebody else. But Joshua, you can also work on the problem. Nothing wrong about it. You please go ahead and calculate. Calculate the F0 value based on the following. D121 is a 3 minutes for a given organism. For a 6 log reduction, calculate the F0 value. Also calculate the F0 value if the Z value is 5 degrees centigrade. So first do the first part, which is tell me the F0 value for a 6 log reduction at 3 minutes. Anybody apart from Joshua who can give me that answer? Anybody who has finished the calculation? F0 value, 6 log reduction, 3 minutes is the D value. Anybody who has worked out the answer, it's such a simple thing. Why has nobody worked out the F0 value so far? It's such a simple thing. Come on, I'm now going to pick up a student who has to give me an answer. Okay, who is Mr. Omkar? Please unmute yourself and can you please give me the answer? Omkar,
Sir, I'm working on it. Still, I'm not getting the.
what? It's such a simple thing. Why you're not getting it? The answer is very, very simple. You've got the data, right? The data is saying what? You need a 6 log reduction. The D value at 121 degrees centigrade is 3 minutes. What will be the F0 value for that?
Sir, is that 18 minutes?
Yes, it is 18 minutes. Who answered that question? Yeah,
Priya.
Priya. Yeah, that's correct. Why, Omkar, are you having a problem in getting that answer? What was the issue? Where were you confused? Omkar?
Yes, sir.
Hmm.
Sir, in log, I was confused.
The log is already told that you want to do a 6 log reduction. So your log reduction value is 6 is already given to you and 3 minutes is the time. So this is a 1 minute question. Just by looking at data, somebody should have jumped and said is this is 18 minutes. You don't need anything. Okay, now Omkar, you will have to solve the next part of it. When the Z is 5 degrees centigrade, what will be the F0 value if I change the Z by 5 degrees centigrade on the positive side? What is Z value? What is the definition of a Z value? Tell me that first. What is Z value, Omkar?
Change in the temperature which will reduce the D value.
Which will
By
achieve the same log reduction of 1 for a D value, correct?
D value. Yes,
So
sir.
now what is the Z value that I have given you? 5 degrees centigrade. What does that imply? If you want to achieve a 6 log reduction and the Z value is 5 degrees centigrade, what is the temperature for getting that 6 log reduction at the same time? Omkar. Z is 5 degrees centigrade, correct?
Sir, 30.
30 degrees centigrade, you are going to go up. So if you increase the temperature by 30 degrees centigrade, okay, you can achieve the same log time in a lower F0 value, correct?
Yes, sir.
Clear? Clear or not clear?
Yes, sir. Cleared, sir.
Now I'm going to share my screen and give you the third problem. Okay, delta T is 3 minutes. T is 132 degrees centigrade. Z is 10 minutes. Calculate F0 using the formula F0 is equal to delta T into 10 to the power of T minus 121.2 divided by Z. I've given you the formula, I've given you the values. All you need to do is calculate the F0 value. So delta T is 3 minutes. T is 132 degrees centigrade. Z is 10 minutes. F0 value is how much? Okay. Calculate this and I will ask a student and they have to answer this. So I'll pick a student from the group and answer it so that I know that the concept has been understood. I'm going to give you 5 minutes to solve. So 12:05, I'm going to pick up a student to answer the question. student. Aditi Pandey.
Sir, ma'am, few of them have gone for photo shoots, sir.
Sir,
3 members.
Aditi Vishnupriya, Shashank and Lasya went for the photo shoot.
Okay, fine. So then Anina Shaju. Can you give me the answer to this question?
Sir, I think it is 36.068 minute.
Correct. Very good. It is 36.08 minutes. That's the right answer. Good. So now I'm going to throw this session open for questions on this calculation. If any one of you have any doubts, I'll clarify that. And then with that, we can end the session for today on this topic. The formulas are there, the calculations are there, but the concepts, if there is any question, please ask the questions.
So, yeah, for the initial question, we use the uh, method of um log no log n0 minus log nt. So, why we didn't we use the FO for formulas? Sir?
Because you didn't know the Z value there, and you were not trying to get two different temperatures. That look, the actual temperature was not, you were fixing the temperature. You were saying that I know that I'm going to do this at 121 degrees centigrade itself, right? So I had fixed the temperature, and for that given temperature, if I want to do a log reduction, what is the FO value which I'm getting? So that was the formula that we used to arrive at the F0 value there, right? So we just took the data at 121 degrees centigrade and we said, if I want a reduction of 6 logs, what should be my F0 value? Okay. This is the theoretical calculation of F0 value that you have done. That was the first example that we did. Okay. When we did the last example where we got 36 minutes, okay, in that situation, we actually took the data of the autoclave. And what is the formula for when we have the actual data? This
Delta
is
T.
T. T, no, what is the T? T that I'm looking at is the actual temperature or the temperature at the time the product was under sterilization, right? So I know that this was at 125 degrees centigrade, for example, or 132 degrees centigrade in this case of an example that we took. At 132, I got, when I put different probes in the autoclave and I set the temperature, I set it at 132. When I did it at 132 and my Z value was known at 10 degrees centigrade as per the example, then and I know that the time interval between two temperature readings. So I took a temperature reading at minute number 1 and I took the next temperature reading at minute number 4. So the delta T of the two readings was 3 minutes. But so I knew that my temperature, I was doing an actual measurement, and at that actual measurement, my F0 was so much. Why is this important? Is that when you are actually carrying out an autoclave cycle, you are not going to be able to determine the log reduction online, correct? Because for you to determine the log reduction on the spore count, you have to take the swab and plate it. Now you're not going to be able to do that real time. You are just going to do the initial load and the final load, and you will see whether your reduction happened or not. So when you are doing in an autoclave, when you are trying to get into a calculation of an F0 online, and I remember I told you that in the autoclave cycle, I will program in the software that F0 value should get calculated automatically by the program. What is the program doing? The program is measuring the actual temperature T. The delta T is being measured because it is calculating that F0 value between a specified time interval. So I can say calculate F0 value every 5 minutes. So it will take the reading at 0 minutes. It will take the reading at 5th minute. If my sterilization cycle was, say, for going on for 45 minutes, it will calculate F0 for every 5-minute interval. Okay. In that, what is the only coefficient which has to be constant? Z has to be constant. So I have to feed Z into the system and say that for this particular organism that I know, in this case, it could be Bacillus stereothermophilus, my Z value is so many degrees centigrade. To get me 1 log reduction, I need 10 degrees centigrade, for example, or I need 5 degrees centigrade, for example. In whatever be that case, I will feed that value based on a known flora into the autoclave program and say, I am fixing the Z value. Now at an interval of 5 minutes each, measuring the actual temperature which was inside the autoclave, tell me what is my F0 value. Okay. And that F0 value, I will compare it with the theoretical value and make sure that at no given instance, my F0 value is lower than that F0 value which was calculated theoretically. That is the use of this information or these two formulas in practicality. So let me go back to the example, right? So in the example, I calculated the F0 value knowing that I had Bacillus stereothermophilus, and I was aiming for a reduction of, let's say, 12 log. If I was aiming for a reduction of 12 log at 2 minutes as a D1, okay, if I was doing at 132 degrees centigrade, at 2 minutes and a 12 log reduction, within 24 minutes, I should get a 12 log reduction, correct? This is the theoretical value. Now, when I did the F0 calculation on the autoclave and I took the temperature every 5 minutes there, knowing that my Z value was a fixed value of, let's say, 10 minutes, if my F0 value fell below 24 minutes, it would mean I have a problem inside the autoclave. Understood?
Yes, sir.
This is the significance of using two different formulas. Good question. Why did I use two different formulas? The first formula which you used was based on actual spore reduction, knowing the data of D and calculating a theoretical value. Whereas the second formula is what you will use on a day-to-day basis on a autoclave practically by looking at the data which is generated from the autoclave by measuring the temperature at different times. And if the F0 theoretical value is higher than the instantaneous F0 value that you measured in a given time interval, then you have a problem inside the autoclave. The sterilization cycle has to be investigated. Understood?
Yes, sir. Thank you, sir.
Okay. Any other questions? Okay. So if there are no other questions, then I'm going to end the session here for the autoclave part. I'm now going to see you on 10th of April, where we will take one last topic on autoclaves, which will be the autoclave cycle development and autoclave cycle validation, where again the concept of F0, D, and Z will be used, and certain other parameters will be discussed. So I'll do that on the 10th of April. And after that, we have other lectures on 16th and 17th of April, which are on different topics. So with that, I would like to end this session here. And thank you for your attention and wishing you a happy Easter, which is coming up. And I hope you had a good Holy festival before you join this session. Thank you.
Thank Thank you,
sir.
sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, everyone. We'll meet tomorrow.
Right. Thank you.